Okay, so we've just spoken about the bare bones anatomy that'll get you through knowing what EEG is, and we're going to do the exact same thing just for the physiology now. So in this video, we're going to cover concepts such as resting membrane potential, action potentials, and graded potentials, which are all super important in thinking about what we're actually measuring with EEG. So neurons, like all other cells, are surrounded by a phospholipid bilayer that we call the plasma membrane. Now, this is acting like a barrier between the interior and the exterior of the cell. We don't want stuff getting into the cell that we don't want inside the cell. And a good rule of thumb to think about what's getting through and what's not is anything large or anything charged is not getting into the cell. But we're talking about excitable cells here. We're talking about neurons. They need to conduct electricity. So we need charged things to get in. And the way that this is going to happen is via proteins that will sit across the plasma membrane and will let selectively ions in and out of the cell. Now, the types of ion channels that we have are resting ones. What these are, they'll stay open all the time and essentially will just allow an ion to flow down its concentration gradient in and out of the cell as it so pleases. There are voltage-gated sodium channels. Now, these will open in response to a change in membrane potential. We have ligand-gated channels. These will open in response to an extracellular messenger of some kind. And in the case of the neuron, it'll be a neurotransmitter. And for completion's sake, we've also got these signal-gated channels. These will open in response to an intracellular signal of some sort. So resting membrane potential really just is a difference in potential between the interior and the exterior of the cell. And in a neuron, this sits at about negative 70 millivolts. So when a neuron is there, it's at rest and it's ready to undergo some form of stimulation. But how does that number come about? Well, the first thing to consider is that we've got these leaky channels open. And in cells, there's a lot more potassium inside the cell than there is outside of the cell. And if we've got these leaky channels open that are permeable to potassium, well, it's going to flow down its concentration gradient outside of the cell. So we have more potassium in here, less out here. The potassium wants to exit, and so it will. Now, every time a potassium ion leaves the interior of the cell, it's taking its positive charge with it. So if we've got positive charges leaving the interior of the cell, well, this is going to become a lot more negative. And in fact, if potassium was the only ion that dictated membrane potential, we would reach equilibrium at about negative 90 millivolts. But we just said that the resting potential of a neuron is negative 70, so clearly potassium is not the only player in this game. The other thing that dictates potential is the sodium-potassium ATPase pump. Now, what this does is it expends energy in order to have ions flow against their concentration gradient. Now, what this does is shuttles 2 potassiums into the cell and then ditches 3 sodiums out of the cell. And what this does is it brings sodium in here, and therefore, as the potential of sodium, the equilibrium potential of sodium is a lot more positive than it is for potassium, the membrane potential at rest will be at about negative 70. So we've got both the leaky potassium channels and this pump dictating that resting membrane potential. So again, before an action potential or before the neuron becomes stimulated, it's sitting at negative 70 millivolts because of the leaky potassium channels and the sodium-potassium ATPase pump. But we spoke about the fact that there's these ligand-gated channels that will open in response to a neurotransmitter. So in this case, let's say that an excitatory neurotransmitter has come along, such as glutamate. It will bind to this receptor, and then the ligand-gated channel will open. Now, in the case of this being an excitatory neurotransmitter, we'll have sodium enter the cell. Now, the addition of sodium inside the cell is going to make the membrane potential more positive, and it will increase slightly. If there is enough sodium inside the cell to move the membrane potential towards negative 55 millivolts, we will hit our threshold to begin an action potential. But if a bit of sodium comes in but not quite enough to get to that threshold, we'll get what's called a failed initiation, and the membrane potential will return back to rest. And action potential is an all or nothing response. Once you've hit the threshold, you've got to commit to it. So let's say that that's happened. Enough sodium has flown in now that the threshold is at we've hit the threshold of negative 55 millivolts. Now what's going to happen here is because we've had a change in membrane potential from negative 70 to negative 55, these voltage-gated channels are going to open. And if these voltage-gated channels are sodium channels, we're going to get the influx of sodium into the axon. Now this here is called depolarization, and this will continue until we get to about positive 40 millivolts. At positive 40 though, the sodium channels will close, and what we'll get is an opening of the voltage-gated potassium channels instead. And what this is going to cause is a flux out of the cell for the potassium ions, and if we're thinking about all of these potassium ions leaving, they're taking their positive charge with it, and the cell will become more negative. In fact, it'll become so negative that we're going to reach this hyperpolarized state in which the membrane potential has now dipped all the way down below resting potential. And this is what we call a refractory period. So the neuron is going to find it really hard to respond to anything now because it's so far away from its threshold for an action potential. Over time though, we'll get the closing of those the potassium gated channels. We know that we've got no sodium gated channels open at the moment. And we'll have that sodium potassium pump come back into action, and the leaky potassium channels will eventually lead to this neuron reaching its resting state again. All of that happens at about 5 milliseconds, and pretty much everything we do is dictated just by action potential. Every time we feel something or sense something, it's really just sodium flowing into a cell, potassium flowing out. So there's one to add to the existential crisis for you. In terms of thinking about how these actually travel down an axon, well, there's two ways that it can occur, depending if myelin is present or not. So if myelin is present, we spoke about how this is an electrical insulator. And what this allows the depolarization to do is essentially spread at these nodes of Ranvier. So we're going to have the action potential regenerating here, here, here. That'll speed up the rate of travel. However, if we have an unmyelinated axon, we'll have continuous conduction, and what this will be is just one segment of the axon will depolarize and then it will repolarize, and then we'll move on and the same thing will happen, and this will just continue until you reach the axon terminals. So we know that neurons don't touch. They need to communicate via what we call the synaptic cleft. So when two neurons come together, we've got the synapse, and let's assume that the action potential has made its way down the axon terminals. So there is a way for this neuron to now release its neurotransmitters. And what happens here is we've got our presynaptic cell here and the neurotransmitter sitting in the vesicle. We've got our synaptic cleft, and then we've got our postsynaptic cell. So what happens here is that the action potential will reach the axon terminal of the presynaptic neuron. When this happens, we'll get the opening of these voltage-gated calcium channels. And if these channels open, calcium is going to flow into the cell. Now, it's the calcium influx that actually triggers the release of the neurotransmitters. So the vesicles will come to the surface, and undergoing exocytosis, the neurotransmitter will fly into the synaptic cleft. We'll then get the binding of this neurotransmitter to the postsynaptic cell at the receptors, and depending on the neurotransmitter present, the postsynaptic cell will either go on to perform an action potential or it won't. And that's what we call a postsynaptic potential. So let's assume we've got an excitatory synapse here. So this is our presynaptic neuron, and it's released glutamate into the synapse. This glutamate will travel and bind to the dendrites of the postsynaptic cell. And if it's excitatory, remember we've got these ligand-gated sodium channels here. These will open in response to the neurotransmitter, and we'll get sodium flowing into the cell. If sodium is in the cell, well, now our membrane potential is more positive. And with enough of this sodium entering the cell, we will get depolarization occurring. So that's what we call an excitatory postsynaptic potential. If, however, we're at an inhibitory synapse where the presynaptic neuron has released an inhibitory neurotransmitter into the synapse, such as GABA, GABA will diffuse across and bind to the dendrites of the postsynaptic neuron. It will open ligand-gated chloride channels. Now, chloride is a negatively charged ion. If more negative ions flow into the cell, it's going to make the membrane potential more negative. And what will happen here is that the neuron will be pushed further away from the threshold and it will become hyperpolarized. Now, each of these mechanisms are what we call a graded potential. So postsynaptic potentials are a graded potential that occur in the dendrites of a neuron that has just received a neurotransmitter. These postsynaptic potentials aren't just something that, one little thing flows across and it's all over. They summate over both space and time. So when we talk about spatial summation, what we mean is that postsynaptic potentials will add together if they occur at different locations, but at the same time. And temporal summation means that over the same location, but at different times, the postsynaptic potentials will, again, add together. So in the case here, we've got a neuron at rest, and it's just received a excitatory postsynaptic potential, and then it's received another one, but it's still not quite at the threshold of negative 55 yet. Then we've had an IPSP come in, which brings the membrane potential a bit more negative, and then another EPSP, slightly positive again, another IPSP, slightly down, and then we've had 2 EPSPs in a row. Finally, this neuron has now reached the threshold, and it will undergo an action potential. That there pretty much makes the basis of what we're going to be measuring using EEG, so we'll speak about this in a lot more detail in the next video.